Hello, bachcho. Let's see the question. So the question says PCR proceeds in three distinct steps governed by temperature, and they are in the order of. So the key concept in this question is to firstly understand what is PCR. So this is moderately difficult, and PCR actually stands for polymerase chain reaction. So PCR is a method of DNA amplification. So we can copy the DNA and make multiple copies of this DNA. And this is known as DNA amplification, which is done with the help of PCR. Now, PCR is a thermal cycle. That is, with each step, there is a change in the temperature. Or we can actually say that due to the change in temperature, there are different steps that takes place during PCR. So there is a thermal cycle which takes place during PCR. Now, if we see the different steps of PCR, then the first step in PCR is denaturation. So this step takes place at a temperature of around 94 degrees centigrade. Now, in this step, there is the separation of the two DNA strands of the DNA template. So the DNA template whose copies we want to make, we first need to separate the two strands of that DNA so that we can replicate each strand and make copies of that DNA template. So therefore, the first step in PCR is the denaturation, that is the separation of the two strands of the DNA. Now, the second step of PCR is annealing. So for the PCR to take place, there are certain primers present. Now this annealing process takes place at a temperature of 55 degrees centigrade. So therefore for annealing process to take place, the temperature in the PCR machine needs to be reduced from 94 degrees centigrade to 55 degrees centigrade. Now annealing means that the primers, which is corresponding to the genes which we want to amplify. So these primers will anneal to the DNA template at their respective positions where they have complementary sequences with the separated strands present in the DNA template. So there are two primers. One is the forward primer, which is attached to the one DNA strand, and the next one is the reverse primer, which is attached to the other end of the DNA strand. Now the third step in this process is extension or synthesis step. So this step takes place at a temperature of 72 degrees centigrade. So a specific enzyme known as Taq polymerase is responsible for the elongation of the primer by the addition of nucleotides complementary to the DNA strand or the DNA template strand. So, Taq polymerase is a thermostable enzyme which has a higher optimum temperature, and therefore it is particularly used in the PCR cycle. So now if we see, then the order of the steps in PCR is first denaturation, then annealing, and the third one is extension or the synthesis of the primer. So now if we see the So now if we see the options, then the first option is denaturation, annealing, and synthesis. So this is the correct order because first we denature the DNA template, then we anneal the primer, and the last one is synthesis, that is addition of nucleotides to the primer or the elongation of the primer. Now the second is synthesis, annealing, and denaturation is also the wrong order. The third one is annealing, synthesis, and denaturation. Again, this is the wrong order. And the fourth one is denaturation, synthesis, and annealing. And again, this is also the wrong order. So therefore, all the three options, that is second, third, and fourth, are wrong because they have different orders given. But the correct option is option number 1, because the correct order of sequence in the PCR cycle is denaturation, annealing, and synthesis. So this is the correct order and this is the final answer. Hope you understood this well. Best of luck. Thank you.